Today we're going to talk about pregnancy, the endocrinology, not all the physiology of pregnancy, because much of the physiology of pregnancy, things that change like in the cardiovascular system or in the renal system or in the respiratory system, understanding all of that requires the other A&P course. So I'm not going to get into it. But we are going to talk about pregnancy, the endocrine changes in pregnancy, some of the physical changes in pregnancy, and the endocrinology associated with lactation and breastfeeding, because it's a pretty interesting story. Remember your practical quiz tomorrow or Friday, depending. Schedule is on an email or on Moodle. The final exam is on Tuesday, starting at 9 a.m. Let me know if you have any issues with that. It's mostly endocrine, but there will be some spot checks from the previous sections of the course. And some of the endocrine questions sort of stand on your understanding of the previous sections of the course, so that they're semi-integrated. All right, let's go ahead and get started. So this was a slide that you saw at the end of the class on Monday. Let's just get oriented. So we have levels of LH and FSH plotted here during the follicular phase and the luteal phase of a menstrual cycle. We have cartoons showing the development of the follicle, from the primary follicle, the one that has won the race with all the primordial follicles, the dominant follicle. The others have undergone a treatment. Some of the others that have started to develop don't continue to develop. Most of them just stay quiescent until the next cycle, or the next cycle, or the next cycle. Antrum, oocyte, zona pellucida, granulosa cells, theca cells on the outside. You could embellish this figure with detail. Here's a mature follicle with the antrum that is rich in estrogens. Oocyte. And then ovulation and development of the corpus luteum. This area here is illustrating, cartooning, the proliferation of the uterine endometrium, and then the degeneration and menstrual flow in the case of no pregnancy, and the degeneration of the corpus luteum. And these lines here are cartooning estrogen and progesterone changes. Remember, low estrogen is what mediates negative feedback and a stable and low LH and FSH. But as estrogen increases, there's a transition to positive feedback. Positive feedback loop, sort of like the action potential, if you want to use that as an analogy. The action potential happens in milliseconds. This happens over the period of days. But so the time scale is completely different. But the positive cycle, so positive feedback, is analogous. More estrogen, more FSH and LH, more development and more conversion to estrogen, more estrogen. Positive feedback until we get to the point where the LH and the FSH actually surge, and that's the trigger for ovulation. If there is no fertilization, there will be no implantation into the uterine endometrium and then menstrual flow. If, on the other hand, there is fertilization, which we will talk about in a few minutes, and development of the embryo, 4-cell embryo and then a multi-cell embryo and then implantation into the uterine endometrium, the uterine endometrium is not just sustaining the embryo, it releases chorionic gonadotropin, a hormone. into the bloodstream. And that chorionic gonadotropin there are the most important receptors for that are in the corpus luteum, and it sustains the corpus luteum so that it sustains throughout pregnancy. There is no degeneration of the corpus luteum. So because we have a sustained corpus luteum, chorionic gonadotropin sustaining the corpus luteum, we have high levels of estrogen and progesterone, which are going to do things that are important for lactation, as well as for pregnancy, in just a few minutes. But the progesterone returns the LH and the FSH levels to negative feedback, to a stable negative feedback control. And so we have high levels of estrogen and progesterone, and you would say, wait, high estrogen was associated with positive feedback here. Why is high estrogen not associated with positive feedback here? And the answer is progesterone. So there is pregnancy, implantation of the embryo develops, the corpus luteum produces lots of progesterone and estrogen, and that's pregnancy. That's the start of pregnancy. This up here, I guess I didn't start there, but that's just a cartoon of the pituitary, right? So there's the anterior pituitary producing the LH and FSH. I'm not exactly clear why that got put into the cartoon, but there it is. Okay. So this is a good slide to go through because, question? Yeah. The chorionic gonadotropin is released from the uterine endometrium. Online people, I should have checked the sound check. Are you okay with the sound? Okay, that's a good sign. Thank you. So let's look at fertilization. Right? There's no pregnancy without fertilization. With ovulation. There is no pregnancy without ovulation, as well as fertilization. Ovulation caused by the LH and FSH surge is a release of the oocyte and some granulosa cells, and we now call that the corona radiata. The oocyte and the corona radiata are ejected from the ovary, picked up by the fimbria, and then migrate, not just flow, but migrate, roll their way down the fallopian tube. The oocyte is viable in the fallopian tube for at least 24 hours and sometimes more. The oocyte is typically fertilized in the fallopian tube, but then implants in the uterine endometrium in a normal pregnancy. So if there is, in the case of fertilization, obviously sperm is delivered into the vagina. The vaginal secretions, the lubrications of the vagina are a little acidic, and the acidic environment of the vagina has both negative and positive consequences for the sperm. The negative consequences is that it kills them off. So that is an obstacle to sperm longevity. In addition, but we'll see in a moment, that the acidic environment is also important for sperm to do what they need to do, which is fertilization. So it's a yin and yang thing. It's a positive and negative thing. Negative is that the acidic environment is, hazardous to the sperm. The other obstacles to fertilization are a mucus plug at the cervix. The cervix produces mucus. Those of you who've taken the other AMP course know what I could be playing here with regard to mucus, but we'll leave that to the other AMP course. Mucus is a secretion. It's a mucopolysaccharide. The cervix secretes it. The mucus blocks the opening to the uterus. It actually changes the LH and FSH surge along with the increase in estrogen, changes the properties of the mucus. It goes from very thick and gooey to somewhat less thick and gooey. Those of you with the other A&P course know that we have salivary glands that produce either a dry, sort of a thick saliva or a watery saliva. Same transition happens in the cervix, and so the sperm can get through the less gooey mucus easier. But they both have some blocking capability to prevent the sperm from getting to the oocyte. There are some white blood cells, there is some immune reaction, both within the uterus. Well, yeah, mostly within the uterus. So 300 million sperm are ejaculated. Half of them go up the wrong fallopian, so some of them don't make it. The ones that do make it, half of them go up the wrong way, right? They don't know any better. Approximately 2,000 out of this 300 million reach the oocyte, typically in the fallopian tube. And it can take hours to fertilize. You'll see in a moment that you may have heard that it only takes one sperm for fertilization. That's not right. It takes thousands of sperm, but only one happy, successful sperm gets to do its job. Typically only one. And the sperm, once it gets past the sort of hazards of the acidic environment of the vagina and the mucus block, gets into the uterus and the fallopian tube, it's viable for several days there. So the sperm can be swimming around in there waiting for the oocyte to meet it. Now let's talk about what the acidic environment does that is positive for this whole story. The surface membrane of the sperm, so this is the head of the sperm, there's a motor and the tail. Those of you who've studied anything regarding bacterial motility with a flagellum know that there's basically an ATP-driven rotor here that spins the flagellum and causes the sperm to move, swim. So, but the head is where the haploid DNA is. The flagellum is the propulsion mechanism. The midpiece is the motor. The surface membrane, let's just focus on the head. The surface membrane of the head is rich in cholesterol, so it's a very stable membrane. But when it's in the vagina, the acid secretions there help remove at least some of that cholesterol. So sort of, right? The acid destabilizes the surface membrane. If it destabilizes it too much, the sperm doesn't survive. If it destabilizes it not enough, the sperm is not likely to be able to fertilize. You'll see in a minute. So there's this Goldilocks, destabilization of the surface membrane, just the right amount, enough for the sperm to survive, and enough for it to be capable of fertilization. When the sperm makes contact with the corona radiata of the ovulated oocyte, there's going to be a reaction in the acrosome of the sperm, this specialized submembrane, it's an organelle of the sperm, and there are enzymes here that are going to be, so it's under the cholesterol, but outside of the cytoplasm. And there are enzymes here that are going to be essential for fertilization. We'll see that in the next slide. So here's a cartoon from a book that from a different physiology textbook, showing fertilization. So you should annotate this slide, which is in your handout. This is fertilization. So here, let's get oriented. These orange dots are supposed to be the granulosa cells of the corona radiata. This purple line is cartooning the zona pellucida between the corona radiata and the oocyte membrane. And then there is the surface membrane of the oocyte. So here's a zoom in of this structure, right? Cytoplasm. Well, there's cytoplasm. There's going to be a surface membrane here somewhere. I guess it's not very clear. I guess this orange squiggly line is the surface membrane. There's an extracellular matrix, just like there is in most cells. There is the zona pellucida, a membrane on the outside of this. And then these orange things are the granulosa cells of the corona radiata. The sperm, are the purple, blue things, obviously. So, what do we see here? When a sperm makes contact with the granulosa cells, triggers the acrosomal reaction, and the acrosome basically dumps its contents, and its contents contain enzymes that help sort of break down the corona radiata. And if the sperm is sort of already through the corona radiata and starting to make contact with the zona pellucida, those enzymes will start to break down the zona pellucida. So we have basically proteolytic and carbohydrate hydrolyzing enzymes released by the sperm acrosome to work its way through the corona radiata and the zona pellucida. One sperm does not have enough of these enzymes to do all of it. But, one sperm will get some of the way through. The next one will get some of the way through, right? Using its enzymes. So they're sort of, each sperm is, they aren't smart, they're not helping each other, but they are in fact helping each other, so that one of them gets all the way through and fuses with the surface membrane, right? One is not enough, and yet one is enough to do this, right? I hope you see the difference with what I'm saying about one not being enough. We need many to do this so that one can do this. Fuse with the surface membrane. The fusion of the sperm with the surface membrane of the oocyte, having gotten through the corona radiata and the zona pellucida, triggers a reaction called the cortical reaction. So the oocyte has a cortex. Cortex just means the surface, really, or the rind. Remember, the cerebral cortex or the cortex of the adrenal is the outer layer. So the cell here has an outer layer, and this sperm getting through, even before it dumps its DNA into the cytoplasm, because that's what we need to make we need to make a diploid cell, right? The haploid sperm and the haploid oocyte genes need to fuse and make a diploid cell, and then we're going to get an embryo. But before that, the cortical reaction, it's an interesting thing. It's a calcium wave that triggers release of the contents of these cortical granules that surround the oocyte. So on the inside of the cytoplasm, on the inside of the surface membrane, there is granules. The sperm triggers this wave of, it's not depolarization, but this wave of bursting out of the cortical granules. Exocytosis really, kind of the cortical granules. And the content of those granules changes the extracellular space and the remaining zona pellucida, changes it so that sperm can no longer get through. Because we want one and not two sperm to get through. So once there is one sperm gets through, does this fusion, detect, the cortical granules detect it, there's this wave of release of cortical granule contents, which, so one granule releases, and it triggers its neighbors to release. And it triggers a change in the properties of the extracellular matrix and the zona pellucida. And then those others nearby, they release, and they trigger the release of others nearby. That's the wave. And so the entire zona pellucida and extracellular space changes in properties and becomes impenetrable. And so that blocks polyspermy. Polyspermy is the fancy name for fertilization by more than one sperm. The sperm is then drawn into the oocyte by, cytoskeleton. That triggers the second meiotic reaction. Remember, those oocytes had gone through meiosis 1, but then they were waiting for this happy day to get fertilized, and now they undergo meiosis 2. We have a true haploid DNA, true haploid cell, well, two haploid cells fusing to make a diploid, stem cell, for all of development. So this one cell now has the potential to develop everything in a human being. And now we're talking about development, which is a whole other course, which I'm not going to do. No way. Are we clear on this, on these interesting steps associated with fertilization, the cortical reaction, and the block of polyspermy? Yeah.
Are the cortical granules just specific vesicles?
Yeah, they are vesicles. They're like, I guess you could think that they were like the vesicles of a neuron waiting to be secreted. I mean, they're like that in general, but not in specifics. They're vesicles lined up, oriented right at the surface, ready to be burst. They don't actually fuse. They don't fuse with the surface membrane. They just burst and change. Well, let's see. Maybe they do exocytose. At least some of the contents of these cortical granules. I guess in order for the contents to get in contact with the zona pellucida, there must be a wave of exocytosis. Yeah, that's what it is. So yes, Tessa, there are many cells that are highly specialized for secretion, and they have secretory granules, and that's what these are. Not exactly vesicles, but similar to vesicles. Hannah?
Do all the vesicles, is it just one vesicle that bursts that triggers the entire, zona pellucida to harden or to not allow sperm to come through, or is it like a trigger?
It's a trigger. So one cortical valve, the fusion and exocytosis of one cortical granule then triggers the fusion and exocytosis of its neighbors, which triggers its neighbors, which triggers its neighbors, and so there's this spread of exocytosis and release of the contents of the cortical granules throughout the entire membrane. So it's not just one, it's all of them, but it was triggered by one.
Is it possible for polyspermy to occur?
Oh, yeah, it is. So, sure, we can get polyspermy. There are people who are triple X, XXX, or XXY, or XYY. That's one of the ways to get those unusual genetic genotypes. It's rare. Often, when polyspermy does happen, the embryo is not viable, and the woman may never even know she was pregnant even for a moment. So here is a flow chart that really, this is the flow chart that tells the same story. So this is the same story. Let's just go through it one more time. sperm binding, acrosome reaction, sperm plural sperm who try to get through the zona pellucida. One gets through all the way. That triggers the cortical reaction, the release of cortical granules, secretory vesicles, enzymes in the zona pellucida and in the extracellular matrix, block polyspermy. The sperm is drawn through cytokinesis, cytoskeleton into the egg, triggering second meiotic reaction, fusion of the two nuclei, and fusion of the genomes to make a diploid cell. And then enzymes to help with all that. And we have the beginning of the embryo. So here is the here's the oocyte, sperm, fertilization, which we've just been talking about, sperm nucleus, egg nucleus. Right now we have a true haploid nucleus of the oocyte. When it went through meiosis 2, it did not generate two cells. Well, it generated two cells, but one of them is the oocyte, and the other is that other polar body, which is just sort of a vestige of meiosis. Two nuclei, fusion, all that good CGI stuff. Mitosis, cell, cytokinesis, cell development, cell proliferation.
You said earlier that when the sperm and the egg cell fuse, it's two haploid cells make one diploid sperm cell?
No,
No.
make a diploid embryo. So we're now talking about a zygote or an embryo. The sperm is always haploid. So there's the haploid sperm nucleus. There's the haploid egg nucleus. Fusion of the nuclei, all kinds of mitosis, cell division. So zygote, a one-cell embryo, two-celled embryo, 4, 8, 16, development of the cell. Of the embryo. How something as simple as this becomes a baby is, it's an amazing story of, development, human, what's the word? Morphogenic as well as other forms of development. This one cell has the capability to become this semi-complicated structure, not very complicated, but this semi-complicated structure has all the programs it needs to develop into a baby. Another question?
Yeah, just kind of a follow-up. That seems like incredibly complex, but you said it comes from that one cell at the very beginning. So I'm wondering if you had a sperm cell and you gave it the right genetic code, could it also differentiate into a person? Or is it very specific to sperm and egg cells?
You need the diploid. Now, if you found a way, what you're talking about is cloning. If you found a way to take a sperm cell and make it diploid with another sperm cell, that's not really cloning, because those two sperm cells, remember in meiosis, each cell has its own random sorting of the genes. So what I just described isn't cloning, but it's taking the DNA from the male, but then you'd have a YY human, and I don't think that's viable. The X chromosome is a real chromosome. That is to say, of course the Y chromosome is a real chromosome, but the X chromosome expresses a lot of genes that must, and some of them must be necessary for life.
So I guess then you would need an egg cell with a copy of that genetic information, and then you would need another possibly stem cell with that with the genetic information from maybe a sperm cell or another egg cell, and then you have to get that haploid.
So yes, you need two haploid cells.
Okay.
And the easiest way to get haploid cells is just to get sperm. Get one egg and one sperm. But I suppose, in the 21st century, who knows what we can start to develop.
Well,
Yeah,
what about like a 1X chromosome? Is that viable?
that's a good question, and I don't know the answer to that question. I think it's not, but I'm speculating, so I don't really know. I don't know if there is a human, no, I don't think so, because that's haploid. I don't think that can develop into a viable zygote. But I'm not sure. Maybe if you fed as a haploid oocyte with all the things that the sperm does other than the gene, you might be able to get that thing to develop. It seems unlikely. I really don't know, but it seems unlikely. Anyhow, let's get back to fertilization and pregnancy. This, multicellular structure. I won't ask you the names of to differentiate. This is developmental biology, going from the morula to the blastocyst to a complicated blastocyst. This is developmental biology. The point is there's implantation into the uterine endometrium. That's the trigger for chorionic gonadotropin, and that's the trigger for maintenance of the corpus luteum inside the ovary. Remember, the corpus luteum is your source of progesterone, which is necessary for continued development. So this is an image of the uterine endometrium. Well, I think it's the uterine endometrium. Maybe it's an image of the corpus luteum. In any case, progesterone from the corpus luteum sustains the corpus luteum, and HCG from the endometrium sustains the corpus luteum. Corpus luteum secretes progesterone, which maintains the uterine endometrium and permits further development. So it's a little bit of a positive feedback cycle there. HCG, corpus luteum, progesterone, sustaining the uterine endometrium, which now could secrete more HCG. So this blastocyst implants in the uterine endometrium, and the cells develop. The fetus now, and the amniotic sac and the amniotic fluid are all fetal in origin. They're not maternal. So remember, this fetus, this organism, has a different genome from the mother in which this uterus which it is implanted in. So we have an amnion and a fetus and an umbilicus to provide nutrients from the from the uterus to the developing embryo, and to allow some of the waste products to be removed into the maternal circulation. And that's what's illustrated here. So let's get oriented. Here is maternal circulation. This pink area here with these squiggly lines is supposed to be uterine endometrium. Here's the fetal umbilical cord, umbilicus. This is fetal circulation. So fetal circulation, maternal circulation, uterine endometrium. The fetal circulation develops these complicated structures called chorionic villi. Those of you who've taken the other AMP will sort of realize, okay, that's a little bit like the complicated circulation in the lung, right? Where the circulation surrounds lung pulmonary alveoli to do gas exchange. Well, here we have fetal circulation in contact with a pool of maternal blood. So there is blood flow through the uterine endometrium into these sort of pockets of blood, which then get circulated back into the maternal circulation. But here, there is contact between fetal blood and maternal blood for the exchange of gases and nutrients. When I say gases, those of you who've taken A&P2 know that the gases that I'm talking about are oxygen and carbon dioxide, right? We need oxygen for oxidative phosphorylation, which the fetus is doing a lot of during development to make ATP for development, and getting rid of the CO2, which is the product of metabolism. That's happening here. So once upon a time, so some of you may have heard of, genetic screening, right? Being able to determine the genotype of a developing embryo. So I'll just take a break here for a minute. When my daughter was in my wife's uterus, when my wife was pregnant with my daughter, who is now 35 years old, we did genetic screening. We through amniocentesis. So, and amniocentesis it was a fairly routine, but a somewhat risky procedure in which a long needle was injected through the maternal skin and uterus into the amniotic fluid to draw out a little bit of fluid. It wasn't the fluid they wanted, they just wanted some of these fetal cells, which they then looked at the genome of to make sure. Basically, it was checking for genetic abnormalities at a pretty crude level. But now you can do, basically a whole genome analysis. A lot has changed in 35 years. So you can sample the amnion to get fetal cells. You can also sample the chorion to get fetal blood cells. So there is now a procedure called chorionic villus sampling. And it is also somewhat risky because you're getting a probe or a needle of some kind into a chorion. And there's always a risk when you're doing something invasive like that. These days, they don't do any of that. They can just sample maternal, because there is some exchange of fetal blood cells into the maternal blood. It's incredible now, but they can just sample maternal blood, and from maternal blood isolate non-maternal blood cells. And the only way to get non-maternal blood cells into maternal blood, the most likely way is for it to be from the fetus. So they can find those 1 in a zillion fetal blood cells in maternal blood of a pregnant woman and do the genetic analysis there. So that's much, much less invasive. Yeah.
So there's one thing I was a bit confused about. How does the immune system of the person hosting the child, how does that not recognize the kid as a foreign body and just destroy it?
Ask Peter Blair, because that's a really good question. The placenta is the placenta is immunoprivileged. There's something about the placenta that tells the maternal immune system, don't kill me. And I don't know what it is. Yeah.
How early can they
So let's see. I'm not an expert on this, but my understanding is that amniocentesis can happen after about 15 weeks. So that's 15 weeks of pregnancy. So that's you know, end of the first trimester, or even beginning of the second trimester. So the first trimester is maybe 13 weeks. So a pregnancy is 40 weeks. So 40 is hard to divide by 3, but 39 is easy to divide by 3, so 13, 13, 13. So you have to get into the second trimester to do amniocentesis, which is pretty late. Because if there is a genetic abnormality, I mean, you know, why would you want to sample this at all? Well, the decision is, if there is a major genetic abnormality, perhaps you terminate the pregnancy, you undergo an abortion. That's the decision of the mother. At least in most places in this country, that's the decision of the mother. Right? So I'm getting into an ethical issue here related to abortion, which is not the topic of this course. But that would be the only reason to get genetic screening, is to know, well, no, that's not true. To know whether the baby is genetically typical is information that could be reassuring to a couple or to a mother. Anyway, probably 14 weeks for amniocentesis, early second trimester. My understanding is that chorionic villus sampling can happen earlier, more like 10 to 12 weeks. So now we're talking end of the first trimester. And my understanding is that the maternal blood sampling can be done as early as 10 weeks. So early in the first, mid first trimester. So obviously in this case, the quality of the information isn't essential, but earlier is better. All right, pregnancy. So we have a fetus developing in a uterus. Amniotic fluid, amniotic sac, placenta. Didn't use that word very clearly. This is the placenta making the contact between the uterine endometrium and the fetal circulation. So, here's endocrinology now. Estrogen, progesterone, and chorionic gonadotropin in green. So, what triggered this? Implantation. Implantation triggers the uterine endometrium, to release chorionic gonadotropin. So this event is implantation, not fertilization, implantation. Implantation, a spike of chorionic gonadotropin. This is presumably on different scales. There's no scale bar here. Chorionic gonadotropin can't possibly be as high, in milligrams per milliliter. It can't possibly be as high as estrogen and progesterone, I don't think. But this is all normalized, right? But there's a spike in chorionic gonadotropin upon implantation, and then it falls off to a steady, but not zero level. So it's this spike where this steady non-zero level of chorionic gonadotropin in maternal blood. That's the blood test for pregnancy. Those little strips are basically a Western blot for chorionic gonadotropin. Because the presence of chorionic gonadotropin is a really good marker for pregnancy. And those things you can buy in the pharmacy now, those strips, are very sensitive Western blot-like things. They have antibodies, there's technology, but they basically use antibodies to chorionic gonadotropin and a color reaction to tell the person whether they're pregnant. And they don't even have to use blood, you can use urine, because some of that chorionic gonadotropin gets from the blood into urine. Chorionic gonadotropin, corpus luteum, production of progesterone. LH and FSH levels are negative feedback, stable control. Estrogen and progesterone can rise slowly throughout pregnancy and then fall dramatically after delivery. So this is, until here, we have pregnancy. let's take a break now. Let's take a 5-minute break, and we'll get into this story of the end of pregnancy, the last month of pregnancy. Let's take a break.